objectives for this set of notes. And those objectives are, number one, what is the basic structure of a virus? Some of that will be review if you had the chance to do that Virus Explorer over the weekend. Number two, how does a virus work? And number three, what can we learn from viruses? And even how are they useful? Okay. So 3 objectives. So let's start with that first one. So when we talk about viruses, basic structure is that there are 2 things that all viruses have, and then there's a third thing that some viruses have. So that basic structure includes a nucleic acid, right, their genome, that can either be DNA or RNA. And as you learned in that Virus Explorer, that some viruses, they have double-stranded, or DS, DNA. Some have single-stranded, or SS. And same with the RNA viruses. You have double-stranded and single-stranded RNA viruses that exist out there. So that's the first thing. Every virus has some kind of nucleic acid within it. Okay. The second thing that every virus has is something called a capsid. And this is a protein coat that typically goes around the nucleic acid. Right, so you have a capsid, nucleic acid, and then sometimes, depending on the virus, we'll put a little asterisk here, sometimes they'll have something called an envelope, which is a lipid bilayer membrane. So some actually, similar to a cell, they'll have a lipid bilayer that encompass the capsid and the nucleic acid, kind of creating this bubble. Okay. So if we look at 2 really common examples, one example is something called a phage. A phage virus or a bacteriophage. Right, in a phage virus, it's going to contain those first 2. Every virus has a nucleic acid and a capsid. And so a phage looks like this. So it has this 3-dimensional (3D is always really hard for me). 3-dimensional, there we go. 3-dimensional, I think it's a dodecahedron head. And this is the capsid. So that capsid is actually made up of proteins, and inside of that you have your nucleic acid. All right, so they store their nucleic acid inside this capsid. And then they actually have this kind of alien-looking thing. that looks like this. And what this is kind of broken up into two pieces, but this is known as the tail. So this is the head, and this is the tail. And there are these spikes, and there's the sheath, and they're also made up of proteins primarily, that tail. So that's what a bacteriophage looks like. And what it tends to do when it infects a bacteria, let's draw a little bacteria here. This is not to size. Generally, phages are super small in comparison to a bacteria, but just for space in our notes, if this is a bacteria, they'll actually kind of mount onto the bacteria with those spiky legs, and they will literally inject that DNA or nucleic acid into the bacteria, which is kind of crazy. And then we'll talk about what it does later, but that's the strategy of a phage. Okay? We'll come back to that question. Wenji asked, aren't some phage good for you? There are phages that are useful that we'll talk about, but a phage is kind of like the probably the most understood and simple structures of a virus that have a capsid and nucleic acid, but it's probably the most commonly talked about in bio. The other type of virus is something, we'll use coronavirus. This could also be something like HIV or the influenza virus. They all kind of fall into this other category where they have an envelope. But you have this structure where this is your lipid bilayer or your envelope. Okay? And then inside you actually have what's known as your capsid, right? So this is protein, this is lipids, and that capsid, it protects the nucleic acid inside. So similar, except these types of viruses have that envelope. Okay. And typically what you have on the lipid bilayer is just like in humans and organisms, we have like receptors and proteins and things in our lipid bilayer. So do these viruses. So they typically have these little proteins, which are important for their ability to infect, which we'll talk about later. But we have these membrane proteins. Right. Now, why this is particularly useful for a virus, if they have an envelope, is because now we learned about how phages infect. These types of viruses with envelopes, they tend to infect in a little bit of a different way. So if we take a cell, let's use, this isn't necessarily a bacteria, it could be a bacteria, but just a cell. And I'm going to draw a mini version of this. Alright, so this is our mini version. That, because it has a lipid bilayer, around it, what this allows it to do is that cells have a lipid bilayer around them as well. So instead of it just injecting, what do you think it's going to do? It's going to fuse with the membrane and then dump its contents inside. So it'll look something like this. So if we had here so it fuses with that membrane. Right? We also have a membrane on the cell here. It fuses and dumps all of its components inside of the cell. You also get elements of the capsid in there as well, right? So it's a little bit more efficient, right? It actually just fuses, and then when it replicates, it's actually going to be able to use the membrane of the host cell to fuse out and go into new viruses. So, two different types, one with an envelope, one without. Okay. So, we've got basic structure. Okay. Let's talk about how does a virus work. We've already started to talk about this. So generally, how does a virus work? So there's kind of four pieces to this. So the first thing it does is that it is injecting or fusing the host cell and dumping its nucleic acid into the host. All right, so we saw with both of this, the genetic material is put into the host. Then, number two, that cell, it has machinery that we know about, and that virus is going to use the cell machinery, and we're mainly talking about enzymes here, to make copies of its nucleic acid. It's going to start turning out copies of its own genome using the enzymes in that host cell. Third, it's going to start to transcribe and translate the genes found within the viral nucleic acid to make. If you think about what was contained within a virus, right, it has to make things like more capsid proteins, which are encoded by the genes of the genome, of the viral genome, or other necessary proteins, like the ones on the envelope proteins, like these. They have to make more copies of those. And then it's got copies of its nucleic acid, it's got all the proteins it needs, and the last thing here is that, number 4, new viruses form. They actually self-assemble. Those capsids come together around nucleic acid copies, and they actually burst or lyse the host cell. And those new viruses go and infect other cells. Okay. So Mr. Anderson is going to talk a little bit more about this bursting, this lysing. And then tomorrow, first thing, we're going to talk about that third objective, right? How we can use these, what can we learn from these. So once you have that down, thank you for your hard work today. Can you have somebody drop off your conversation notes at table 5 familiar? And we'll pick this up tomorrow.